Hello, everyone, and welcome to my poster on analyzing the spatial heterogeneity of tumor mutation burden and immune infiltrates on whole slide images and how that signals correlations with bladder cancer survival. First, a little bit of background. What is TMB, tumor mutation burden? It's the number of somatic mutations divided by the length of sequence queried in megabases. It's usually calculated from whole exome sequencing, and it's a promising biomarker for immunotherapy in diverse cancers, including bladder cancer. Muscle-invasive bladder cancer is a type of cancer that has pretty low survival rate, high relapse rates, and the current biomarkers that are being used have low response rates. The problem with tumor mutation burden is that it's sequence-based, which is expensive and time-consuming, and it does not really account for the complexity of the tumor microenvironment. So why are we looking at images? Well, the standard of care for diagnosing cancer is actually pathology slides. So they're basically ubiquitous, and considering that bladder cancer is such a heterogeneous disease, we can use these images to capture the morphological features of the tumor microenvironment. And there's been work that has shown that the spatial colocalization of tumor regions and immune cells has actually associations with clinical outcomes in other cancers. So our hypothesis is that whole slide image analysis methods can capture the morphological features of cancer tissue with high tumor mutation burden, and then their spatial distribution and the colocalization with tumor infiltrating lymphocytes can inform patient outcomes. To do this, we have 3 different data sets, starting with the Cancer Genome Atlas bladder cancer cohort, which was collected prospectively from treatment-naive patients for the most part. And the slides are from fresh frozen tissue. Last one is a study that is also prospective, but mostly with refractory patients, and the slides are FFPE. And the Cleveland Clinic cohort CCF was retrospectively collected with refractory patients as well, and FFPE images. Okay, moving on to the methods. First, we performed tumor detection using a convolutional neural network based on the VGG16 architecture and trained on pathologist annotated whole slide images. Then we take the binary mask of the tumor regions and divide them into non-overlapping tiles of 256 by 256 microns and calculate local binary pattern features on which we perform affinity propagation clustering. From there, we select the exemplars of these clusters and use those as representative tiles, which allows us to reduce our computational costs. We then use these representative tiles and perform transfer learning using an exception model trained on ImageNet. And we take the last layer before the classifier and perform principal component analysis. Then we use that 100-dimensional vector to train a support vector machine model, both on linear and Gaussian kernels, using the TCGA dataset, labeling TMB high patients using the 66th percentile and TMB low with the 33rd percentile. We obtain 2 outputs, that is the tile level TMB prediction and the patient level TMB prediction. For each tile level, we assign the values of the corresponding representative tile to the rest of the tiles in its cluster. And for patient level, we took the weighted average of the representative tile features, weighted by the size of the cluster. Here we can see that we tried different approaches, and our proposed method performed very well compared to other convolutional neural networks and different versions of our methodology. We tried validating our model with our BLAST data set. However, using the same thresholds, we ended up with a pretty imbalanced data set towards TMB high. We need to keep in mind that it's a small data set and it is biased. So once we have our model, we can calculate the Shannon entropy of the tile level predictions to produce a score for the spatial heterogeneity of tumor mutation burden. And here we can see that slides that are ground truth TMB high can be predicted TMB high, and when they have low heterogeneity, most of the tissue has similar values. And when it is predicted with high heterogeneity, we see that there's a high variety of predicted TMB values. We try to see if patient level tumor mutation burden alone was able to discriminate survival, but that wasn't the case. But once we combined TMB prediction with spatial heterogeneity, we were able to see a significant difference in survival. Beyond that, we took a subset of patients from TCGA that were known to have high TMB based on sequencing data. And we observed that our predicted TMB high patients with low heterogeneity had an increased significant survival over the rest. Then we predicted lymphocyte infiltration using a ResNet model trained on a publicly available data set of pan-cancer tumor-infiltrating lymphocytes and determined TIL-high patients to be the ones that had the ratio of lymphocyte tiles to tumor tiles to be higher than the median. We then looked at how including tumor-infiltrating lymphocytes affected survival, and we see that patients with TMB high, low heterogeneity, and high predicted lymphocyte infiltration had improved survival compared to the others, including TMB high, heterogeneity low, and low TILs. Then we proceeded to validate our findings with our Cleveland Clinic cohort. However, we did not see a significant survival difference. And in fact, we see a trend of low TIL patients having improved survival. So in conclusion, we were able to predict TMB using only whole slide images. We identified that spatial heterogeneity of TMB could be a factor in survival outcomes, at least in TCGA. We found that image-based prediction was able to segregate patient survival beyond what sequencing alone methods could do. We identified that there's a potential interaction between lymphocytes and tumor mutation burden. And finally, tumor mutation burden is generally only considered as a single value, but maybe considering its spatial characteristics could be a valuable approach for prognosis. Of course, there's limitations to our work. Our Cleveland Clinic and our BLAST1 cohorts may not necessarily reflect the TCGA cohorts since they have later stage disease, more metastatic disease, multiple lines of treatment, more variation in the size of the tissue, and also the tissue was preserved in a different manner. Also, our TMB validation cohort is fairly small and heavily TMB high skewed. So we definitely need larger, better annotated, multi-institute and more variable data sets with diverse stages of disease for improving our training and validation so we can improve our performance. With that, thank you very much. Please put your questions down in the comments and I will do my best to answer them.